So good afternoon, everybody. Thank you for joining us. We are still waiting for a few more participants to this webinar to arrive, but in the meantime, welcome to this CooperSurgical webinar. Today we will be discussing the sperm separation and methodology and the scientific evidence for the ZyMōt multi-sperm separation device. We will be joined by Neelke de Munck in a minute. She is a paid consultant for CooperSurgical, and she will be expressing her personal opinions and perspectives. So, my name is Martine. I am the Director of Professional Education at CooperSurgical. I am an embryologist for over 30 years, and I joined CooperSurgical in this role roughly 8 years ago. And my role is to create, to deliver, and to coordinate professional education, evidence-based knowledge sharing with our customers. And I'm really pleased to have this role. So I'm also very happy to be your moderator today. And thank you for inviting me, Marcel and Simon. So today we will be joined by Neelke de Munck. Neelke de Munck is the lab director at the Brussels IVF Center in Belgium. She is a highly accomplished PhD graduate with an extensive experience in embryology IVF, as well as also a prolific academic contributor. She has been working as a clinical embryologist since 2010, and she initially focused on oocyte vitrification, and that was also the core topic of her PhD. In 2018, she joined IVRMA, and she became the scientific advisor for the lab in Abu Dhabi, and she was leading embryologist over there as well. In 2021, she returned to Brussels IVF in Belgium, and she became the head of this large IVF lab, Brussels IVF. Her main focus now is to optimize efficiency and increase the quality of IVF and ICSI, etc., all these procedures offered to their patients. So for this webinar, we invite you to, during the presentations, we invite you to put down some questions, and please use the Q&A button, and you can write your questions in that box. You can also upgrade a question. If somebody else asks a question, you can then confirm that you like this question. That will avoid you from asking the same question. And then when I'm doing the Q&A session with Neelke, I will then take those questions with the highest grade first. So these are a little bit our rules and regulations for the webinar. So let's give you a brief introduction to the ZyMōt multi-sperm separation device. So what is it and what does it do? It is a sperm separation device. It comes in two sizes. We have an 850 microliter one and a 3 ml one. And it separates sperms based on their motility. It is easier to use, it's more reproducible, and it gives better sperm quality post-preparation than a standard swim-up, as being demonstrated by several studies. So how does it work? Well, the device, it consists of two chambers, a lower chamber and an upper chamber. We will, between those two chambers, there is a permeable membrane that has small pores. These are 8 micrometers in size. We will inject sperm in the lower chamber of the device, and we will add medium to the higher chamber of the upper chamber of the device, and then the sperms will swim through the pores into the upper chamber, where we will then collect those very, very motile sperms. There is an increase in motility that has been observed in studies, those sperms arriving in that upper chamber. So that is the principle of this sperm separation device. How does it work in practice? Of course, you still need to do the liquefaction of the semen sample. Then you will aspirate the semen sample in a 1 ml needle, and with this syringe, you will then load 850 microliters on the device in the inlet pore. We will then drop little drops of medium, in total 750 microliters of medium on top in the upper chamber, and then we will cover the device, place it in the incubator, and leave it for 30 minutes at 37 degrees. The sperms will do their work. They will swim to the upper chamber, hopefully, and then we will collect from the outlet port with a fresh syringe a sample of 500 microliters, which we can wash like a standard washing procedure in your centrifuge. And then it is ready to be used for IVF, ICSI, and IUI. So does it really work? And I'm going to quickly go through a few of the data that we know that are out there, the studies that have been done. There are some great studies that give us really interesting information. The first one is a study by a Swedish group of the team of Adolfsson, and they looked at split ejaculate, prospective randomized study, split ejaculates. One part was prepped with the standard density gradient centrifugation, the other part with the ZyMōt, and what they could see was that there was an increase in motility, more motile sperms as compared to the density gradient sample. And the concentration, however, of the sperms that were motile was lower than with the density gradient centrifugation. And here you can see the individual sperm samples. There were 25 in this validation study. And the light blue bars are the ZyMōt prepped. This is the progressive motile sperms. And in all the samples, this was significantly higher in motility. That was the observation that they made. Another study, this is the Parella group, they looked at DNA fragmentation, again comparing density gradient with the sperm separation device, the ZyMōt, and they could see that there was a significant decrease in sperms that were carrying DNA fragmentation. So this was the initial 20.7% of the sperms having DNA fragmentation, and after ZyMōt prep it was only 1.8, while compared to density gradient there was 12.5. Another study looked at the outcomes in the clinic lab itself, the quality of the embryos, etc. And this is the group of Bunty. They found significantly more blastocysts, significantly better quality blastocysts, so their utilization rate was significantly higher than when using the density gradient. And surprisingly also, there was a significant increase in euploid embryos. Other studies looked at the euploidy rates as well using ZyMōt devices, and here you can see that there was clearly a trend. So again, the pink purplish columns, they are the ZyMōt samples. There is a trend, and lots of them are significantly different, positive, that you have more euploid embryos. There's also this great work by the team, as I mentioned before, from Neelke, the Munck's group, and they worked on DNA fragmentation, for example. And I'm inviting now Neelke to share her screen. I'll stop sharing, and if you could share and then present your data to us, that would be ever so great. So please, if you have questions while she's prepping her screen, please submit those in the chat, and then we will address those after her presentation. So Neelke, the floor is yours.
Yes, thank you, Martine, for the nice introduction, and also for already explaining a little bit the ZyMōt device. So as the title of the webinar is about the ZyMōt, I will today talk to you about our experience with the sperm separation device, but also give you a little bit of background on studies that have been performed with the ZyMōt device. So, as Martine already mentioned, I'm a consultant for Cooper. Just to guide you a little bit on who I am and in which center I am working. So I'm working at Brussels IVF. For the ones who don't know it, it's in little small Belgium, also the capital of Brussels. We do quite some pickups and inseminations, so it also means that we have quite a lot of sperm samples. So if we just look over the last 4 years, the total pickups that we have done, you see that we are quite stable, we're a little bit close to 6,000 samples on a daily basis. If we then look which sperm samples are being used in these cases, we see that in fact, show you a little bit better, we see that in most of the cases, a fresh ejaculate is used to treat those pickups. So these are, of course, the major source of samples that would be eligible for the use of ZyMōt. If we then look at the number of transfers on a daily basis, we see that on average we have every day around 20 transfers, so it's quite a lot. And if we look at the inseminations, whether it is with donor sperm or with sperm from the husband, we see that there are also quite a lot every month. So we are quite a busy center, which means that we are, of course, constantly looking for optimizations, and especially if they also optimize efficiency and time. So from that perspective, ZyMōt was of highly interest to us. So Martine already explained a little bit, so I will not spend too much time on it, but still repeat a little bit. So there are two different devices. We have the 850 device and the 3 ml device. They both have the inlet in which the liquefied sperm will be added. Then you have the membrane and you have the outlet where after 30 minutes the sperm will be extracted. So here you can see it from the side. You have the inlet and then the sperm, of course, will move because it's a separation based on motility. And you will see the membrane that the sperm will need to pass, and it's only the sperm that is able to pass this membrane that in the end can be extracted from the outlet chamber. So if we look at those two devices, the 850 device, of course, can load 850 microliters, and in the outlet you can extract 500 microliters, which is different from the 3 ml device where we insert 3 milliliters, and at the outlet we can take 1 to 1.5 milliliters. So how does it work? Here you see the syringe with the liquefied sperm, which will be inserted into the inlet, and the 850 microliters of sperm will gradually move through the device. It will be topped with a HEPA solution, and then we put it in the incubator. It's an incubator, 37 degrees, and during 30 minutes incubation, the sperm separation will take place. And then, of course, you see how the sperm moves. So this is the inlet chamber. The sperm starts to move. You see the membrane and the motile sperm that is passing through the pores of the membrane. And then in the outlet chamber, only the best selected and separated sperms will be present, so they can be aspirated at the end of the procedure after the 30 minutes. Just to give you a closer look what those pores of the membrane look like, this is a close image of the pores through which the sperm cells will swim. So what in fact do we know already about the use of ZyMōt? In the meantime, there are already quite a lot of abstracts published and also publications. So I will give you a little bit an outline of things that I think are important for you to know. So it's a comparison of centrifugation, or in general density gradient centrifugation. What do we know about DNA fragmentation? Is there an effect of reactive oxygen species? What is the cellular stress? Is there an effect on fertilization and blastulation? Also the ProID, which was already highlighted also by Martine. What do we know about outcomes? And then also important, can it be used in IVF? So this is a paper that was published some time ago, where they in fact checked washed samples, stream-up samples, and then samples that pass different types of pores. As mentioned by Martine, we used the 8 micrometer pores, but in this study they also tested 3 micrometer and 5 micrometer pores, and they looked at the generation of ROS, so the reactive oxygen species. And what you can see is that whenever you use a sperm separation device, irrespective of the pore type, compared to a ROS in the sample, you see that the production of ROS is really reduced compared to a washed sample or a swim-up sample. In this study, they also looked at the DNA fragmentation, and yeah, of course, as we know, a raw semen sample always gives high DNA fragmentation. But when we look at SWIRAP sample and the different types of pores, we see that there is really a reduction in the DNA fragmentation index, and specifically lowest in the 8 micrometer pores. So they conclude a reduction in ROS as well as a reduction in the DNA fragmentation index. Another publication, it's an abstract on the use of the microfluidics device to select sperm with lower DNA damage, and also to look at the motility. So they specifically compared the ZyMōt device to the conventional ZyMōt device. And in summary, they conclude that the device is able to select sperm with a lower DNA fragmentation and also a higher motility when they compare to the ZyMōt. So here we can see the results. The sperm DNA fragmentation index in ZyMōt was only 10%, or it was 20% for the ZyMōt group. And then also they looked at the blastocyst rate. There was no significant difference, but it was a little bit higher in the ZyMōt group. So 53% in the ZyMōt and 46% in the ZyMōt group. Then in this trial, they also compared the ZyMōt device to a density gradient. And they also looked at fertilization and embryo quality. So you see here the control, which is the density gradient, and the microfluidic sperm sorting. You see fertilization rates are similar, not significantly different. When they look at the high quality embryos on day 3, they see 66% gradient, 68% for the microfluidics, so it's higher in this case. When they look then at the rate of the blastocyst, they see that there is no difference in blastocyst development between both groups. So this is a good difference. They also looked at clinical data. So again, comparing density gradient to the microfluidic device. And as you can see, all p-values are above 0.05. So it means that the clinical pregnancy and the ongoing pregnancy rates for transfer or patient are always the same between both groups. So again, reassuring data, similar cleavage and blastocyst quality, and also similar clinical and ongoing pregnancy outcomes. What else do we know? The ploidy was already mentioned also by Martine. So in this abstract, they looked at the microfluidics device and they checked how it would be able to increase the ploidy rates compared to density gradient centrifugation. So what you can see here is the ZyMōt device and the density gradient, and you can see the number of 2 PNs as 7.3 and 6.8. So there was no difference. Then if they look at the mean number of blastocysts, you see that in the ZyMōt device, significantly higher number of blastocysts was obtained compared to the density gradient. And then when they analyze the total number of embryos, you see that it's also significantly higher in embryos generated with sperm from the ZyMōt device. So this was also the trend that Martine showed earlier. So in this case, not only improved the eukaryote rate, but you can also see improved blastocyst rate, 10% higher in this group. Then also very important is the use of ZyMōt in insemination cycles, because of course you not only want to use it for patients coming through the IVF lab, but also insemination. So what they did, they compared again ZyMōt density gradient, and what you can see is that, of course, after the preparation, you will see significant differences. So sperm concentration is known to be a little bit lower after the microfluidic device. This is also partly because the fact that we only treat a specific part of the sample, the 850 microliters, while of course for a gradient, we will use a higher volume, which means that you can also select more sperm samples. But then, very important, if we look at the motility, you can see that the ZyMōt device is in fact able to to have almost a pure population of motile sperm, which is also reflected in the total progressive motile sperm count, which in the end is higher in the ZyMōt device, despite the fact that the sperm concentration is so they looked at clinical outcomes comparing this ZyMōt device and the grading device based on these samples. And what you can see is that in fact always the pregnancy, and ongoing pregnancy rates are higher with the microchip device. However, they did not find a significant difference in this case, but still it can be clinically relevant for the study groups. Then we come to our experience in Brussels IVF. So before we start with the clinical implementation, we will of course first test to see how the device is performing. So what we did is we divided one raw semen sample in 4 parts, which was then treated as wash and a density gradient. These are in fact 2 techniques we generally use in our lab, and wash for bad samples, density gradients for better samples. And then we also compared with the MAX system and the microfluidic sperm sorting. MAX was specifically selected, of course, to select against apoptotic cells. So what we analyzed was motility, morphology, the acrosome index, DNA fragmentation, as well as the vitality. So in total, 52 samples were included. You can see the sample characteristics of the unprocessed semen here, also volume of 4.6, concentration 58.9 million, with a good progressive motility of 58%. And then we see the sample after it was split in 4 and treated with a wash, density gradient, the magnetic activated cell sorting, and the sperm separation device. So what is important here is that you see that indeed this progressive motility is indeed significantly higher in the sperm separation device, and also the highest compared to the 3 other techniques. And also important is the DNA fragmentation index, which is significantly lower and the lowest after sperm separation device. So these are all good results depending on the sperm separation. As I mentioned before, we were highly interested to check also the magnetic activated cell sorting, specifically to see how capable that device is to deselect these apoptotic cells, and if the ZyMōt device would be better. And in fact, if you look at the results for the apoptotic cells and the dead cells, you see that in fact it's always on average lower in the ZyMōt device, meaning that that device is in fact more capable of eliminating those dead cells. Then we have another abstract, which was presented at the Alpha Congress. So this was a single oocyte study with a primary endpoint utilization rate. So this study was designed after we confirmed, of course, in this previous trial that indeed a good proportion of sperm is selected in terms of motility and DNA fragmentation. So we required 253 oocytes in each arm, and of course we wanted the ejaculated sperm and at least 6 mature oocytes. Like this, we have 3 and 3 oocytes at least in each arm. So when we look just purely at the male and the female, then we see females were 34 years old, males 37 years old. And when then we compare the characteristics of the sperm after preparation with density gradient or with ZyMōt, and we can see that the concentration is more or less the same, but importantly, the progressive motility is significantly higher in the ZyMōt device. When we then look at the development of those embryos, and you can see here the total number of oocytes assigned finally to each arm. So when we look at a fertilization rate, the normal fertilizations with 2 embryo rate was 78 and 78, so not a difference between both groups. Good quality embryos on day 3, 76 and 77, so also not different. And also on day 5, we could not find any difference in the development, 41 and 36%, excellent and good quality. For us, a very important parameter is always to look at utilization rate. So this is a number of embryos that is finally used for the patient, which can mean or being transferred or being cryopreserved. And you see that there is no difference at all between both groups, whether you look premature side or fertilized. So conclusion from this abstract was that as fertilization and mortality and utilization were similar in both groups, this device in fact offers an advantage of eliminating the negative effects and time-consuming nature of centrifugation. And of course, it allows you to schedule samples in a more efficient way as well. So based on these results, we decided also to start implementing the ZyMōt device in our clinical setting. If we then look at what we win, because now we have a little bit of experience, we checked 1 month just to see what is in fact the time that we win. When we look at the density gradient centrifugation, we see that on average it takes you 39 minutes and 56 seconds, so almost 40 minutes to process one sample. If we look at the ZyMōt device, in fact, if you look at purely the technique, we in fact only gain 1 minute. But this is of course because the sample needs to incubate for 30 minutes, which is of course different for the density gradient centrifugation, where you have multiple steps that you need to run through, and there is not a lot of waiting time. And it's this waiting time in the ZyMōt device that would allow the efficiency or the increased efficiency by in fact treating multiple samples at the same time. Just to give you a few examples of what a raw sample looks like and what the sample will look like after the ZyMōt. So I'll give you 4 examples. So this is a sample with concentration 3.59, so a little bit lower concentration, and you see the motility, okay, 12% A, 34% B. So progressive motile sperm is not that bad. And if you look after ZyMōt, we see concentration of 0.4, which is not too high, but you see that in fact most of the sperm is motile. Another sample, a little bit better, so you see higher concentration and more or less the same motility as the first sample. And you see here that you can already double the concentration above 1 million, and again 97% motile sperm. Then if we look at a little bit better sample, so the concentration 28, motility A at least 50%, and then you see that you really moved up to a really good concentration after ZyMōt with again very high motility. And this sample is even better at 57 concentration, 71% A. You see that the final concentration after ZyMōt is really nice, as well as the motility. So these samples are in fact all perfect to perform peaks. If we then look how we can start in the lab, because that is in the end the question we asked ourselves, are there really benefits of the ZyMōt device? Really, yes. And there is improved motility, there is improved DNA fragmentation, there is no reduced fertilization, no reduced blastocyst development rate, and we see that it gives you an improvement in sperm separation time, as in you generate flexibility to potentially treat multiple samples at the same time. So as I mentioned before, if we look at density gradient, then there are multiple steps which you need to go through, and of course they all require manual handling, which means that you cannot treat another sample while one density gradient centrifugation, which is different from the ZyMōt device. And you perform the sperm analysis as with the density gradient, but then you have a big block incubation time of 30 minutes, followed by the final sperm count with density gradient. And it's in fact these 30 minutes that would allow you to clean your workstation or move to another workstation and start with a second sample. And depending on how smooth the samples enter and how when the samples are and how smooth you work, we would as well be able to treat, in fact, 2 samples or start 2 samples in the time that we would perform one-stage degradant centrifugation. Which patients should you include? And this is always a little bit tricky because one of the most important parameters of the ZyMōt device is, of course, the presence of motile sperm, because you need the motile sperm to pass through the pores. So when we started, we used the data that we had at that moment, which was the data from the sibling oocyte study. And based on this, we saw that the recovery was good for samples with more than 1 million concentration and more than 10% A+B. For frozen samples and IUI, we did not yet do the validation, but I know that there are abstracts out where frozen samples and IUI are already selected. Of course, very important, if you start with this in the lab, it's important after 1 month, after 2 months, just to do validation and see how you are performing compared to the periods you had before you started using this. So how can you integrate it then in the end in your lab? And you need to arrange a schedule for sperm sample preparation, which is of course important if you really want to benefit from buying multiple samples within one time frame. You of course need the samples as well, so there is a chance that you will need to adapt this schedule in which the patients want to be treated. Then also important is the lab setup. It's always important that there is division in time and space. So even if you would be able to treat multiple samples at the same time, maybe that you have a clear working flow to first clean completely your workflow while the sample is in the incubator for 30 minutes, that your flow is completely empty and that you can start your second sample. Ensure that you have an incubator, that you have a good protocol, that all your people are trained. This training can also be provided by CooperSurgical. They have an online tool that you can show to the people and which they can follow with some tips and tricks on how to use the device. And as I mentioned before, always evaluate what you are doing. Do it first in short steps and see that you don't cause a negative effect that you will not want to use. I'll show you here also the picture of the incubator that we are using in the lab because we have this incubator. It means we have also 6 chambers and each chamber can contain 1 patient. We also hang a timer on each of those 6 spots that you can really separate all your patients. We also only have 3 workflows to perform samples for IVF ICSI, which means that every operator in theory could handle 2 samples with the ZyMōt device at the same time. So, very important, keep validating even after you applied that technique and maybe adapt a little bit as you're going to ensure that you don't cause any damaging effect to patients. Thank you.
Thank you so much, Neelke. Very great and beautiful, inspiring presentation. So I'm now looking at the questions. We've got a good set of questions coming in. So first of all, what steps are necessary to start using ZyMōt in the lab? And then the next question linked to that is, what steps are necessary to train the staff on the ZyMōt use? So could you elaborate on this?
Yes, so to start using the device in your lab, I think it's important that first of all, you validate that the device is working in your center. Even if after 1 or 2 trials on a sperm device, you see that it's not working, you can of course always contact the company because maybe you're doing something wrong. But I think an initial validation, more or less like we did, would be a perfect start. Not necessarily to split your sample to 4, but use it or compare it to the technique that you are using currently in your lab. Is it a swimmer? Is it a density gradient? just make sure that you do not cause any harm to the patients, that at least the motility is the same or is going up, similar for morphology and DNA fragmentation, if you perform these tests. And then, a sibling oocyte study is always a good way to see if your patients, or at least if the development and the fertilization is not affected. And it's in fact a very easy study. You can do it on 20 patients, on 30 patients, and we usually go to 50, but it's also because we have quite some patients in our lab. So if you're a smaller clinic, just decide to do it on around 20 patients, and just make sure that this study will help you to define criteria to include patients, in fact, that will be eligible to use for the ZyMōt. I forgot the second question.
No problem. Then,
training.
so you have validated, you find that you have similar or better results. So how then do you transfer this into the lab to the other operators, the other embryologists or lab technicians?
Yeah, so this is always important that you have a good protocol, a good procedure, which is written and explained to the people. And then, as I mentioned before, there is this online training course. I think it's 1 hour that all the people can follow, and they really, it's interactive. And you really need to follow and do as they say, and they will also tell you if you're doing things wrong. So that is a good way to see that, in fact, people use it in a correct way. Irrespective of that, I think also sometimes things can go wrong. I mean, even if we do a density gradient centrifugation of a good sample, sometimes we see that the recovery is not what we expected. And it is possible, of course, that it also happens for the ZyMōt device. But of course, it can also be that you just, maybe did not aspirate in the correct way.
Mm-hmm.
So that's why it's always important, to have a good training. But of course, I mean, there is always chance that people make a small mistake. It's no problem, because you can, in fact, if the recovery will be too low, you can just apply a new device, a new 850 microliters, and wait for a better recovery.
Yeah. So the clear points in handling the device would be good connection with of the syringe with the device, slow injection, slow dropping of medium, making sure it's nicely covered, the membrane is fully covered, covering the device, putting it in the incubator. We forgot the labeling, so we need to make sure that this device has the name of the patient, that it's witnessed and all that, or electronic witnessing tags are added, and then to recuperate, to retract the sample from the outlet port, also do that in a nice and steady way, I'm assuming. Yeah, okay, perfect. So let's see what other questions we have. How about volumes of ejaculates? So what about large volumes? And then, of course, I will ask you, what about small volumes?
So, I mean, big volumes, in theory, not an issue. You apply 850 or the 3 ml as you want. Bigger volumes are usually no issue. I think the most important question is indeed the lower volumes, and they're in fact also no problem, because you can just, if you don't reach the 850 microliters, you can just add the remaining volume that you're missing with the EBUS or the sperm washing solution.
Yeah.
So both are equally the same and equally effective.
Yeah. And so for the large volumes, you only take 850 microliters. That's what I hear from other groups, that they don't need the full sample to be worked with.
No. I mean, there could be an option to maybe concentrate your sample, but the problem is that, of course, then you also take a lot of cells, and maybe then you can block the filter.
Yeah. True. We have a question coming in saying, can you indicate what sperm DNA fragmentation test you used for your study?
Yes, in this case, we used the tunnel staining. Yeah, but if you look in literature, you will see that multiple abstracts and publications are done, can be with a halo test, with a tunnel test. I mean, multiple tests are performed, and
Yeah.
the results are in fact the same for all types of tests that have been performed.
Yeah.
DNA fragmentation is always lower.
Yeah. Okay. And the next question is, can you share your thoughts on why DNA fragmentation will be so much lower in ZyMōt compared to ZyMōt, since it is both of them are motility based?
Yes.
Why? What is the scientific reason behind that?
Well, it's a good question. It's something we did not test ourselves. So I do not have data from myself, but indeed you would expect that the DNA fragmentation would be quite similar for a ZyMōt device compared to a Sperm device. There was one study which I showed you where they checked the reactive oxygen species and the DNA fragmentation index, and in fact there also the Sperm sample had reduced DNA fragmentation. So maybe it depends on how they perform the swim up. Probably, I mean, even though the angle should always be the same and the incubation time also, maybe the way how they aspirate is more prone to differences between operators, which is not the case with the ZyMōt device. So maybe less controlled, which may lead to differences between studies, because I'll tell you there are studies where it's the same and there are studies where it's higher in swim.
Yeah. So then we have a question here. Could we use the ZyMōt on surgically retrieved sperm cells, such like TESE, MESA, PESA? Have you experience in this? Do you know about studies on this?
I don't know about studies on it. I think it's a bit tricky from two perspectives. First of all, motility. Testicular sperm samples do not always have a good concentration of motile sperm. And secondly, there are more cells and bigger cells in testicular samples. So I think the chance to block your pores with a testicular sample will be bigger compared to an ejaculate.
Yeah. But epididymal sperm cells for patients who have had a vasectomy, for example, I think it's all about concentration and motility.
Yeah, indeed. We never tried it. I'm also not aware of a study where they did, but indeed.
No, neither do I. No.
Fine,
but
be used.
Yeah. So that is still, so for those who have asked that question, why don't you start the study and let us know and publish the data? We need to learn from each other. That is very important. I see a raised hand from Takia Yeboah Kingsley. Could you write your question in the Q&A box? That would be so good. Then I can ask Neelke that question. Then there is an interesting question. Does the use of ZyMōt, could it increase issues with sperm capacitation so that they're capacitated too early? And if you use them in IVF, then you have lower fertilization rate?
Well, one of the issues with IVF and the use of the ZyMōt device can be the fact that you still have seminal plasma remnants in your processed sample. And that is also why Martine showed in one of the first slides that for IVF, it's in fact better to perform 5 minutes interrogation after your ZyMōt separation to be sure that all the seminal plasma is gone. And normally this should give good results for IVF. But personally, we in our center have not yet tested the ZyMōt device for IVF.
Yeah. So have you tested any of these embryos using PGTA, so to see if you have more euploid? Did you? Oh, maybe they are going to do it.
And no, we did not do like an RCT or a sibling study, but of course we have started now since the 5th of August, to be very precise, 2024. We started applying the ZyMōt device for all patients with the characteristics that I mentioned, concentration
Mhm.
more than 1 million, motility more than 10% A plus B. So once you have in your first ejaculate these parameters, and you are planned for ICSI, you will your sample will undergo ZyMōt separation. And this is, of course, for patients with PGTA, PGTM, regular ICSI patients. But of course, the whole bulk of oocytes is injected with that sample. So we don't have data to compare this.
Yeah.
Also not in RCT, but I mean, we could check, but then of course you have different populations, different periods that we could check.
Yeah, to set up a split study, a sibling oocyte, so to speak, study for this involves a lot of work, and it's a study, so that needs paperwork, etc. Yeah. Okay. What about viscous sperms, ejaculates? How do you deal with that?
You know, it's no problem. We do not have a lot of viscous samples, but it happens once in a while, and specifically when there were really very viscous samples, I talk to people, you know, when there's a very viscous, call me, even if it's not a patient for IVF ICSI, if it's just a routine sample, let's put it on ZyMōt just to see what the recovery is. And we use up from category 0 to 4 to describe the viscosity. A 4 we never tested, but up until 3 we have tried, and the recovery is very good.
Yeah.
So okay. For viscous samples, it can even be better to perform a ZyMōt compared to density.
Yeah. So you do your standard liquefaction period, and then you don't use the smaller needle system to break down viscosity, or
the smaller needles we use as the very last option. We prefer to do first the gentle pipetting or adding the heatless, and the syringe is really only at the end. But even then, the sample is very viscous. No problem.
But it might be worth that you guys also publish this or have an abstract on this, because we get this question so frequently. What about viscous samples? And well, we can only explain that it works if we have scientific evidence, so to speak. So thank you for sharing that. Okay, then really, really important question. What about viral or bacterial contamination using this system? Do you know of this? Have you tested this? Will it reduce, or do we still have to add an extra system to remove these bacteria/viruses? So I'm thinking about.
It's a very big question, to be very honest, because there are lots of viruses and lots of bacteria.
Yeah, but I'm specifically hepatitis or HIV.
Yes, okay, for hepatitis and hepatitis B, I cannot tell you because we do not have a validated test on the sperm to test the hepatitis B. We asked for it already, even for other samples. We cannot validate it, so I cannot help you on that one. For HIV, we've thought about it, but the number of samples of cases, in fact, that we have is so low that we did not yet explore it. For the if we look at bacteria, I cannot yet disclose the information, but we are running a trial. Okay, so we will hear from that in the future from your team. Perfect. So we dealt with the viscous samples, we dealt with concentration issues, we dealt with, I'm just going through the questions here. Do you have, a good question, do you already have living healthy babies with this technique in your center? I'm calculating, yeah. And from the sibling study, I know we have clinical pregnancies, but I don't know if we check the live birthday, but they should be. So we are using since August, the 5th of August we started. Yeah. Okay. We should have. We will have for sure. But I did not yet look at it specifically. But we have now used it for 10 months. Okay. So we should have the first live. We need to be patient as
The pregnancy takes 9 months. So yeah, that's good. Okay, is there any other thoughts you want to share with us? Because I think we're coming to the end of our Q&A session. Any extra things, tips you have for our...
Just validate and evaluate. That's all I can say. Even when you validate it, revalidate after you use it.
So make sure you have data, that you have traceability of everything you do. That's what you're saying. And then evaluate on a regular basis if this is working. And maybe also check the cutoff value for the concentration of sperms, motile sperm that you use in the ZyMōt compared to the density gradient, because that might be different. And for IUI as well. Yeah. So really look at your data. That is what you are recommending. Okay.
Yeah.
Okay, perfect. So thank you, Neelke. So let me then grab my presentation again.
There's one raised hand, Martine.
Yes, but we've asked them if they could write the question in the chat box. So I'm waiting for that. I can't see it. So that was your presentation. Thank you again so much, Neelke. We now come to the end, and I would like to make a summary of what has been presented today. I think it's clear from your data, from the data out there as well, from other colleagues in the field. It's easier to learn this method than the traditional methods. As you rightly said, there is this video, the interactive method to be trained in using the ZyMōt. When you are trained and you use it, it's easier to use. It has fewer steps, so that could influence the time, although in your hands it was only 1 minute, but you could be freed. Your operators are freed for a longer period to do other tasks. Witnessing is still in place. It could be electronic witnessing with tags or manual witnessing, and there is a reduced risk of errors because there are fewer steps. It is a reproducible technique. It gives consistent results, which you also show clearly with those nice overviews of different samples. And also from your study and others, the sperm DNA fragmentation is clearly way lower when we use the ZyMōt for the sperm separation as compared to the standard density gradient centrifugation techniques. There is a link to aneuploidy, and there are reports that there is an increase in euploids. So I hope that we can keep confirming that increase in euploidy. The scientific reason behind all of that, we are still exploring that as well. So all in all, this ZyMōt system gives a better quality sperm with a faster, easier and preparation method. So there was just one last question. I'll deal with that if you don't mind, Neelke. Can I use a ZyMōt for a very small ejaculate? Yes, you can. You need to add extra medium until you reach that 850 microliters that you will have to load onto the device. So using medium to get and mixing it a little bit before you slowly inject it into the lower chamber, you can use those poor quality samples as well. You can separate sperms from those as well. Okay, so on this note, we got great information, super great information from Neelke de Munck. We also have CooperSurgical are organizing a series of webinars. These are called Expert Insights. And Neelke was in the past one of our speakers, hence we contacted her to speak for you today. But the next Expert Insights, we have invited Eleftherios Gavril and Maria Banti, who will talk about the use of ZyMōt and cryopreserved samples. And Maria Banti specifically will talk about euploidy rates post-ZyMōt use. So please scan the barcode or ask your local distributor to send you an invite to go to the registration platform so you can attend that one as well. The last Expert Insight webinar was on the use of Sage one-step GM-CSF culture and transfer medium. Very interesting as well, in the outcomes there. And that one is now freely available. So if you scan the barcode now, or you ask your distributor to send it to you, or you go to our CooperSurgical Knowledge Hub, you will find this webinar as well. It is really informative on how to use it, what the outcomes are. So you can watch it at any time you like, because it is freely available at this very point. So with this, I would like to thank you all. Thank the expert Neelke again for sharing your knowledge, and thank you all for attending. But I also would like to hand over now to Simon. Simon, where are you? Yeah, there you are. You would like to close the webinar. So I'm handing over to you, and thank you, and see you soon.
So thank you very much, and I'd like to thank all the attendees for coming to this very interesting webinar. I want to thank also Neelke for the very interesting insights on the experience working with ZyMōt at her lab, the Brussels IVF, as well as reflections to other publications. I think it's safe to say that the results from working with the ZyMōt are quite good, and also compared to conventional methods. Thank you a lot to Martine for the moderating role, which was great. And then here at the end of it, I urge you all to contact your local distributor, the one that invited you for the meeting, for additional information. You might want to get your hands on publications, and that can be provided by your distributor. And of course, you also have a chance to find out how to get the ZyMōt. All of that, including price information and other logistics, can be provided from our distributor. So I urge you to contact them as soon as you can. So thank you very much for attending, and see you at another webinar at a later point. Thank you.